Hello, my name is David Chrimes. I'm the Business Development Director for CooperSurgical Fertility and Genomic Solutions, and today I'll be presenting on ERPeak PGT-AI-SM, which is a PGT-A analysis technology, and also the latest evidence for PGT-A. So the topics covered will be ERPeak. Now this is our CooperSurgical endometrial receptivity test, and we will be covering what is endometrial receptivity testing, exactly the Cooper Genomic solution. We will then look at the PGT testing overview, so that's pre-implantation genetic testing. In the overview, looking more deeply at PGT-A through our PGT-AI analysis solution, and then a very quick review of some of the latest evidence for PGT-A and why it's important for patients. So let's start with ERPeak, the endometrial receptivity testing. So in November 2018, Cooper Genomics released our endometrial receptivity test called ERPeak. Now this is a new genomic test for determining endometrial receptivity in IVF patients by measuring the expression of some key hormone regulated genes. And this allows us to accurately determine when the patient's window of implantation should occur. And this targeted approach on gene expression analysis through our custom developed and validated platforms allows a much better targeted for a particular subset of patients undergoing IVF. So why get into endometrial receptivity? Well, there is increasing amounts of literature that for the endometrium receptivity is a good target for the improvement of pregnancy rates, specifically around the patients with repeat implantation failure, RAF patients. And here's just a few snapshots of some papers which have been published over the years. And as you can see, there have been quite a number. And the reason why this has really taken off is that in a lot of mammals and animals, actually the embryo drives a huge amount of the endometrial receptivity. So the embryo is releasing some kind of signaling molecules, allowing that to then drive the implantation of those embryos into the endometrium and ultimately lead to a pregnancy. And in some animals, they can even store these embryos for weeks or even months in a fixed stage before everything comes together. In humans, it's very, very much endometrium driven. So there is a fixed window where the endometrium is ready for the implantation of an embryo. And the embryo is believed has very little signaling and impact on this. So really what we're looking to do here is make sure that when that embryo is transferred, it is transferred at the time when the endometrium is at its optimum condition for embryo implantation. So if we think about the stages of implantation in a natural cycle where there's no IVF, then the egg is fertilized, it then forms into an embryo and is traveling down the oviduct, hatches and enters the uterus at time ready for the time the endometrium is ready to receive that embryo, and it starts the implantation process. And this is controlled by the change in hormone levels from the egg follicle after the egg is released, and it's all timed for that receptivity. Now in IVF cycles, of course, then we are transferring an embryo, these days typically day 5, day 6 embryo, which may or may not have been hatched via a laser, but nonetheless we are transferring this embryo under a fixed protocol with an expectation of that endometrium being in the correct state to receive the embryo. Now, this is very much textbook in terms of process. Now, what we do know is that from an increasing number of studies that not every woman indeed follows the textbook. And so it's now thought that between 25 to 30% of women with repeat implantation failure have a displaced window of implantation. So a woman with repeat implantation failure is considered by different groups slightly differently, but let's say they've had 2 high-quality embryo transfers with not even a pregnancy. Some people say it has to be across multiple cycles, other people just say high-quality embryos. But in any case, for these patient groups, there are a significant percentage of them where they're not getting pregnant, and it's been shown due to the endometrium not being at the right stage for receiving the embryo. So how can we work out the endometrial receptivity status? Well, thankfully there are plenty of women in the world who are keen to help people achieve pregnancy, and they are fertile and they've undergone some research, and what that allows us to do is track the endometrial gene expression at stages of receptivity post release of the egg through a natural cycle and through normal hormone levels. And what that allows us to do is build up gene expression patterns and start clustering these endometriums into samples, and through high-powered computing and cluster analysis, we can start really clustering the stages of the endometrium into non-receptive. These are typically the proliferation stage still. Pre-receptive, i.e. the endometrium is not yet at a stage suitable for receiving an embryo. Receptive, ideal timing for an embryo transfer. Or post-receptive, the endometrium has actually passed its peak and we should be looking to do an earlier embryo transfer. And so by using these, we can then take endometrial biopsy samples from people undergoing IVF and see if their expression profiles match any of these clusters, and then use that information to guide the embryo transfer. So to develop this assay, we undertook a non-inferiority study. So the most validated test on the market for this test is the iGenomics ERA assay, and it has some clinical validation and clinical studies behind it. So what we did is, part of our non-inferiority study, is under full research protocols and full patient consent, endometrium biopsy samples were taken, and that biopsy sample split. So half of it went to iGenomics, half of it came to us, and we compared the results. And we compared the results to both our internal expectation and also to the iGenomics reports. And after AI and machine learning, and we'd run thousands of simulations of our data, we slowly built up the cluster map so that we can see clear differentiation between the phases. And this is really how we built up our validation data set and robustness to the assay. So why do endometrial receptivity testing? Well, in this diagram here, we can see that basically we expect that when we do an embryo transfer, it will be typically 8 to 10 days after the ovulation in a natural cycle, or if it's under a controlled process, 5 days after the start of progesterone treatment. Now, what can happen is this individual can respond differently to the hormones, and the timing of the window of implantation is shifted away from this theoretical in about 30% of women, and by getting this right, we can increase pregnancy rates in these recurrent implantation failures patients. So the typical patients which can benefit from ER testing are the recurrent implantation failure patients, Recently, they've started looking at the adenomyosis patients, and these are patients where the endometrium has actually started to grow through the uterus wall, muscle wall, and it's believed that these patients also have an increased chance for displaced window of implantation. The frequency of these patients has ranged anywhere from 5% upwards in terms of the IVF population, and so they're definitely an area of increased interest. We also have the purely economic example where patients have undergone IVF, they've struggled to get a euploid embryo. Now they have a euploid embryo, they want to make sure that it's transferred absolutely at the right time. And so these, again, are increasing numbers of patients who are embarking on endometrial receptivity testing. For you as an IVF clinic, there's 4 very simple steps. So we ship you the endometrial biopsy and sample shipment kit, and we'll have a look at that in a second. You ship that back to us. We do the laboratory analysis. We report the results. Now, these results are reported within 2 weeks of receipt of the biopsy sample, allowing a transfer in the following cycle. And then you can do a precision embryo transfer. So if we look at the sample collection and shipping kits, we have these kits sent directly to clinics. We have courier provided to you to ship them back to us. So we work with you very closely on the best method for shipping them back to us for testing. But each kit contains your sample collection tube, your biohazard bag, the test requisition form, which also includes a patient consent form, the sample preparation and shipping instructions, and we can also supply you with the endometrial sampling device as part of CooperSurgical. So all of these kits are shipped to you. They have a long shelf life. They can be left on the shelf for when you have a patient, so you don't necessarily have to order them patient by patient. And then we have these test documents. So obviously within each kit, we send you the test document, which reviews how you complete the test requisition form, you perform the endometrial biopsy, and how you ship the samples back to us, the test requisition form, and ultimately you will get your results back from us. So you have 4 test result options really. So we have pre-receptive, receptive, post-receptive, non-receptive, and no results. So the no result is, as the name suggests, we completely fail to generate any kind of data from this test. It is very rare, very low percentage of cases, and these normally come from either the biopsy being too small, very bloody when it was taken, or something, it wasn't put into the biopsy buffer correctly and stored correctly prior to shipping. As I say, our current expectation is that is in a very low percentage of cases. So let's look at the true test result option. So if you're pre-receptive, that means at the time you took the biopsy, the endometrium was 24 hours away from being at its optimum receptivity. So that means you've taken the endometrial biopsy on the day that you would normally have placed the embryo back, and when it comes to the embryo transfer, instead of doing it that day, delay it by 1 day. Receptive is obviously you're perfect, you're right in the middle of the receptivity phase. That is a perfect day for doing an embryo transfer. If you're post-receptive, it means that your endometrium has actually gone too far, and you should have done the embryo transfer 1 day before. If you're non-receptive, then you don't match within the 3-day window that we're measuring, and generally these endometriums are still in the proliferation stage. So they would need probably another 2 days before they were ready for the embryo transfer. We can look at that in more detail with you should you get one of those results. Now, as part of our endometrial biopsy test, we don't require any blood work, we don't require any endometrial thickness to feed into the answers to give you these results. We do, however, presume that the person doing the endometrium biopsy has taken to some extent a look at the endometrium and decided that there is nothing fundamentally wrong with that endometrium that would lead them to believe there's an underlying clinical issue, such as large numbers of endometrial polyps or raging endometriosis. So we are assuming that these are patients that basically look as though they have a healthy endometrium, but somehow they are not getting pregnant. So the collateral we provide, the extra paperwork, is we have a lovely patient brochure which allows them to view it in your clinic as to why they may consider this, and we're working on some extra web links to show how we are going to take this forward. So now we're going to move on to PGT and embryo testing. So just to be absolutely clear, there are three forms of PGT, pre-implantation genetic testing of embryos. So the one that we're really going to be focusing on today is PGT-A, the pre-implantation genetic testing for aneuploidy. We also have PGT-M, which is PGT for monogenic disease, classic example, cystic fibrosis, or you have PGT-SR, structural rearrangements, and these are basically translocations and inversions. Now, just to be clear, these names were released in 2017, the back end of 2017, and were formalized through the American Society for Reproductive Medicine, the International Committee for Monitoring Assisted Reproductive Technologies, ICMAT, and also the World Health Organization. And this is to bring standardization, because different people called, say, PGT-A, PGS, some called it CCS, comprehensive chromosome screening, some people called it PGT-A, pre-implantation diagnosis for aneuploidy. This was leading to confusion in the market, therefore it was agreed, these terms were agreed to bring clarity and simplicity to the market. So PGT-A is a genetic test designed to improve IVF outcomes, and it provides information about the chromosomal health of the embryos. So to be clear, this is looking at chromosomal copy number. This is not an overall absolute, this embryo is completely healthy and will result in a completely normal live birth. This is looking exclusively at the chromosomal competency of the embryos. And what it's designed to do is lead to the transfer of the embryos most likely to lead to an ongoing pregnancy. So again, it's not a magic bullet. It is not going to guarantee a healthy live birth, but it's the best shot that we have. So if we look at the history of PGT-A development, it has been known for a long time from looking at product of conception testing that aneuploidy in embryos or in fetuses is the major driver for miscarriage. So the theory has been proposed that if you could test embryos for this aneuploidy before you transferred them, then of course you stand a much higher chance of weeding out the embryos which were likely to result in miscarriage. This theory has been around for a long time. We started in 1990 by looking at fish when that was 3-color, looking at the major live birth trisomers, 21, 18, 13. We then moved to metaphase fish. And in 2008 is really the first time we were able to do an entire 24 chromosome embryo screen from a single cell. So this is looking at every single chromosome within that embryo. And we did it both in polar body, so pre-fertilization, and also a single cell from a day 3 embryo. In 2014, the technology started moving away from arrays and through to next generation sequencing, which is still used today, and we did our first NGS PGT-A case in 2014, but also reciprocal translocation PGT-SR. It's basically a copy number assay, works very well on sequencing. Nothing really changed between 2008 and 2014, and then nothing changed again between 2014 and 2018. And we launched PGT-AI in November 2018. So PGT-A technology via next generation sequencing allows full detection of aneuploidy, that's whole chromosome aneuploidy, partial chromosome aneuploidy or segmental aneuploidy, unbalanced translocations, some forms of polyploidy, so that's things like XXY, XYY, and then our favourite topic, which is still a hot topic, mosaicism. So as technology improves, as everything else improves in terms of embryology, we started detecting stuff that was previously unknown. Now, mosaicism, unlike aneuploidy, is not related to maternal age, but it's a mitotic cell division error at some point during the embryo's development. So it's fertilized normally, it has then gone on and had a mitotic error. And that results in an embryo which is a mixture of both euploid cells, for want of a better description, good cells, and aneuploid cells or abnormal cells. So classically in PGT-A, we started off at day 3, they were either euploid or aneuploid. We now have this mosaic situation. So one of the key questions has always been in a day 5 a trophectoderm analysis, day 5, day 6, how representative is that first biopsy of the inner cell mass, the part of the embryo that goes on to form the baby? Now, thankfully, if the first biopsy is completely aneuploid, then the inner cell mass was always completely aneuploid. In very rare cases, 4% was at any euploidy in any of the other trophectoderm tissues. Again, if it was completely segmental, so a complete copy number change was seen, the inner cell mass was always abnormal. So this shows very clearly if your first biopsy is abnormal, there's almost 100% chance the inner cell mass, the baby, is abnormal. Now, if we start looking at mosaics, a mosaic complex in this case is 3 chromosomes showing a slight variation. If you did another biopsy, in 60% of cases, it was completely euploid. If it was, sorry, 42%. If it was a segmental, it was about 60%, and a mosaic aneuploid, so a single chromosome, you've got about 30%, 30-40% chance of some euploid tissues. But really, it's the inner cell mass, the baby component, that when we start looking at mosaics, it's around 50-50 whether that inner cell mass is normal or not. And that is presumably why there is increasing evidence that mosaic embryos don't do as well as euploid embryos. But basically, whatever your first biopsy is a very good indication as to what the inner cell mass is going to be. So if we look at this work that was presented by an Italian group, Francesco, they looked at mosaicism, and basically if you transfer a low-level mosaic, so below 50%, the outcome is not that dissimilar to a euploid. So the implantation rate is not that greatly different, the miscarriage rate or number of early abortions is similar, your ongoing pregnancy rate is slightly reduced, and the number of babies born is trending below. High-level mosaics, much lower implantation rate, much higher frequency of early abortion, much lower rate of ongoing pregnancies with a much reduced level of live birth. And we found in another paper published by Santi Monet, very, very similar outcomes. Basically, low-level mosaics didn't do that much worse than a euploid. As soon as you start getting multiple aneuploidies or high-level mosaics, then you see a really significant decrease in the likely outcome for that embryo transfer. So we moved to a point of looking at whether mosaicism was at all age-related and if that could be a factor, and we found that actually clinically relevant mosaicism as a percentage of the number of euploids doesn't really change depending on age. So this is not an age-dependent thing. This is presumably a culture effect or some embryology effect. We don't really know, but basically whatever the patient cohort is coming in, they have an equal chance of mosaicism. Now we know mosaicism is there in all age ranges, and we know the outcome. We then move to a position where we really wanted to help the clinics by defining the level of mosaicism into 2 levels, what we call split level reporting. So we have euploid, which has a normal, which has the highest chance of a successful pregnancy. Low level, it's the next nearest. It's not as good as a euploid, but it's pretty good. We're calling it here low. High level mosaic, low unlikely. And once we get to abnormal, then it's very, very low, very, very unlikely. And this is great help for the clinicians. However, if you're going to start recording and splitting out mosaicism in this way, we wanted to be sure that we were doing it highly, highly accurately. And this is where PGT-AI technology comes in. So these are typical NGS results that you would see published of a euploid embryo. At the top we have a female, at the bottom we have a male. If all PGT-A samples look like this, that would be fantastic. Unfortunately, they don't. Now, the embryology lab, the embryologist, has taken a nice day 5, day 6 embryo and taken a portion of cells, 5 to 7 cells off, put those cells into a very small volume of buffer, 2 microliters, frozen them, shipped them to our testing lab where we have undertaken the molecular biology. Now, obviously, embryos also come in varying degrees of quality. And so what can happen is we can end up with profiles where a human is forced to judge. If we look at chromosome 1 and chromosome 2, where the chromosome 1 and chromosome 2, A, different from the baseline, this thick, bright green line here, and a chromosome 1 and chromosome 2, the same level of abnormality. Now, this traditionally would have been looked at by 2 highly trained technicians completely independently. If they both agreed, then great. For a case like this, almost certainly a 3rd senior scientist would look at it again to make sure of that call. And then ultimately, the clinical scientist would have to put their name to the report. Now, if you're the clinical scientist and you have potentially an adverse outcome, then you are most likely to be cautious and advise that this is a mosaic embryo. Now, this is another case for subjective interpretation. If we look at chromosome 6, it's a bit wavy. Is this a low-level mosaic, or is it just noise within the system? And this is our kind of noisy profile, for want of a better description. Of particular concern would be chromosome 20 over here, towards the right of the screen. Is this euploid? Is it aneuploid? Is it noisy? What do we make of it? It's all highly subjective. So the questions we were asking ourselves, which blips are real and how do we know? So 1Q, in this case, it's a translocation case, and 1Q here is deleted, and it's clear to see by eye that it's deleted, but the software we were using would not call it automatically, and the same is true with the 9p duplication. And here we have another translocation on the right with much smaller segments, but again, these weren't called even though they're clearly visible by eye, and neither was this mosaic loss. So again, for PGT-SR, which is still essentially a copy number based test, lots of human interpretation was required. So we wanted to build a much better system, and so we started looking at using artificial intelligence for PGT-A calling, and basically we would still have data generated via NGS, but instead of humans looking at it, use a computer. Now, for those who have ever looked or utilized NIPT, non-invasive prenatal testing, this is all done by computer algorithms. Humans do not look at NIPT data. It's been highly validated, highly trusted, and this is where we wanted to get PGT-A. So transfer decisions are based on big data and lots of information. So our basic requirements were to remove subjectivity, take the human element out of it, standardize calling, doesn't matter who's doing the analysis, same results. This reduces risk both for us as a testing company, but also for the clinics. They know exactly what's happened. And we can increase our signal to noise ratios, and we can establish the truth, and this will give us an overall improved accuracy. So we used PGT-AI. We used AI to develop some innovative technologies for PGT-A. Now, AI is the theory and development of computer systems to perform tasks normally requiring human intelligence, mainly decision-making based on information. Machine learning, so you train the machine what to do using truth data, and then it learns from experience. It can also provide recommended courses of action, but the main thing is it requires big data. Now, we as a testing company provide testing for well over 10,000 embryos a month. We have literally hundreds of thousands of embryos that we have tested over the years, and we could use that big data. Most of us already use machine learning in one way, shape or form, and if we don't, our children certainly do. So anyone with an iPhone may have tried Hey Siri, Android, OK Google, Amazon's Alexa system. All of them use AI. You voice train them, you give them commands, and they give you hopefully what you were asking for. But basically, AI, or artificial intelligence, allows us to make user-independent assessments and for reporting. So really, all I'm trying to say here is mathematics and statistics are trained on a large data set. It removes the human subjectivity. Computers don't get tired. They remember everything they've seen before. They don't carry bias. And this also avoids human errors during interpretation and transcription. So what I want to also really stress is that internally, we will be looking at developing this over time with more outcome data. However, for you, for the testing, these are carefully controlled and validated for clinical use. So our design was to leverage our big data, move to statistics, remove subjectivity, allow automatic calling and transcription of results. This is absolutely key. We have tracked as a company live birth outcomes. So we have reported embryos as euploid, they have been transferred, and we know that they have resulted in a healthy live birth. Therefore, we know that anything we saw in that profile did not impact the outcome of that pregnancy. This turned into being the largest training set ever deployed in PGT-A. So we had 1,000 live births, 659 aneuploid embryos, so embryo samples that we knew were aneuploid not just from PGT-A but other methods such as SNP-based testing, known translocations. So these are great cases for building algorithms because you know exactly where the abnormalities in the genome are. And then we also made mosaic mixtures where you dilute known abnormals and euploids to ensure that you're calling these mosaicisms correctly. Our initial black box, our kind of internal validation data was 10,000 sequenced biopsies. Obviously now, we launched it in November, we've run 80,000 embryos through this. So we now have, what does it mean to you? What does it mean to us? So basically what we've done is built a reference database. So all of these 1,000 embryos DNA were resequenced and we built a baseline, and this gave us well over 5 billion data points. But rather than having a really tight theoretical 2 copy number, we had all of these samples overlaid. And so when you load your true sample over, which is this blue line, above your more complete baseline, which is all these grey lines, you can start using statistics and probability to work out whether this blue line is truly statistically different from anything that has resulted in a live birth, previously reported euploid, or not. And in this case, for this change, no. For this change, absolutely, it's clearly statistically different and needs to be reported. So our artificial neural network was deployed, and you basically, in essence, you run loads of samples and you tell the training set that this is monosomy X, this is a female, this is triple X, this is XXY, XXY69, male XY or XYY, and you keep running it until it can automatically recognize the patterns and start calling these automatically. And this is just an example of part of the artificial neural network for sex calling. In the real world, if we go back to some of these plots, so here we have chromosome 1 and 2 on the left that we were looking at before with PGT-AI, it's clear that chromosome 1 is a low-level mosaic, whereas chromosome 2 is in the boundaries of euploid. Again, chromosome 3, mosaic, chromosome 4, not at all. So we just need to really see the improvement, and this grey smudge here is our baseline now. This dashed line is 20% mosaic. We can really see how PGT-AI has helped clear up this data very, very nicely. And again, this sample that was noisy and we were worried about chromosome 20 is completely euploid. We can say this with confidence. And this allows us to provide much more confident results as to whether these samples are euploid, mosaic, low-level mosaic, or aneuploid. So when we are presenting our results as we are here, there is absolutely no doubt when we give you these results whether they're euploid, low-level mosaic, high-level mosaic, or aneuploid. And what this has allowed us to do is provide the clinics with approximately 3% extra euploids. So by that I mean for every 100 embryos shipped into us, an extra 3 called now definitively as euploid. That should lead to a higher number of embryos available for transfer, which should lead to a higher success rate for embryos tested via PGT-A. What it also leads to is much more confidence between what is a high-level mosaic and an aneuploid. So if it's truly aneuploid, it's going to have a very, very, very low chance of success. And so there's no point transferring it. And again, we're adding extra confidence to our calling. But this is all very well, but ultimately, when the patient comes in, we need evidence for this. So what we have is some overview for some randomized control trials. Now, even though the one at the top, the Massenbrook et al. 2007 has been widely discredited, it keeps being brought up by those who don't believe in PGT-A as a study which clearly shows it doesn't work. This work was very, very flawed. It's been rebutted numerous times, but in the interest of clarity, we've put it here. But what are we looking at? We're looking at improvements in implantation rate, IR, increase in clinical pregnancy rates, and a reduction in miscarriage rates, MR. So all of these show various positive outcomes for PGT-A. The meta-analysis again is trending towards improvements in implantation rate, clinical pregnancy rates, live birth rates, reduction in miscarriage rates, sustained implantation rates. And the good thing is these cover a multitude of technologies. So if we look at delivery rates, now this was actually based on qPCR, a much lower resolution PGT-A than we do now, and it covers all age ranges. We can see in the study group, the PGT-A, they have a much higher clinical implantation rate per embryo transferred, a much more improved sustained implantation rate, which in a sense, means also a reduction in miscarriage, a significant reduction in miscarriage, which ultimately leads to a much, much higher delivery rate per embryo transferred. Now, one of the criticisms of PGT-A has always been that people score it against per embryo transfer and not per cycle started. And so ultimately, what if you transferred everything over multiple treatments? Now, what these two studies clearly show is if you treat with PGT-A, your time in treatment, if we look at the left, is much, much, much reduced if you have PGT-A. So you need to generate more embryos and stay in treatment longer if you transfer every embryo available without PGT-A testing and just go on morphology alone. If you look at the genetically competent embryos, then you get that time in treatment, ultimately that pregnancy leading to a live birth, is much, much reduced with PGT-A. Now, in this other study where they tracked embryos over 3 complete cycles, so they were using PGT-A, this was a big clinic in Australia, and they tracked it over 3 cycles started. And again, for PGT, what they call PGT-A or PGT-A as we would call it now, you reach within 3 cycles a cumulative probability of a 60% or 0.6 probability of a pregnancy reaching a live birth, lot quicker than if you do no testing, you're doing every single embryo transfer. This is real data, this isn't mathematically modeled. And so what we find here is actually PGT-A it reduces the time in treatment. It's also cost effective because we're doing less embryo transfers, less time in treatment. And this is really showing how PGT-A can help patients. And this, again, I would like to point out, is in the older age range. All the women in this trial were above 37. Obviously, when looking at data, we need to make sure we're comparing like with like. The clinic cohort that you have may differ from a clinic down the road. So if all you ever do is very high responders with a high chance of pregnancy, or euploid per embryos tested could be radically different, like in clinic 1 compared to clinic 6, where you may only take or specialize in the low responders, the difficult cases. And so we do need to take care when we're looking at these data to make sure we're comparing like with like. But all the RCTs, I say, came out in favor of PGS or comprehensive chromosome screening for implantation rate, sustained implantation rate, and live birth rate. And if we look at the American Society for Assisted Reproductive Technology, so this is a database where every clinic in the US is requested to submit their pregnancy data. This covers every single technology in use today for PGT-A, so that's PCR based, NGS, this isn't our data, this is truly globally, this is every clinic in the US. We can see that IVF with PGT-A, if we look at miscarriage rates, basically for embryos tested up to the age of about 40, then we can see a significant reduction in miscarriage rates for those that have PGT-A, the green bars, compared to those who don't have PGT-A, the blue bars, and it's pretty stable at around the 13% mark. As the woman ages past 40, then even with PGT-A, there is a slight increase in miscarriage rate, but it's nothing like as marked as the miscarriage rate for those who don't have PGT-A. So we can clearly see here that PGT-A is massively reducing the chance of a miscarriage, irrespective of your age. And if we look at the live birth rates, again, per embryo transfer, but we can see here that if you don't have PGT-A, once you reach past 35, your chance per embryo transfer is much, much reduced. And really, again, this is showing that by using PGT-A we can maintain high pregnancy rates per embryo transfer irrespective of patient age. Yes, as the patient gets past 40, they do suffer some decline in pregnancy rates, and there must be an age effect there, but it's mitigating most of that age effect. And so the published benefits of PGT-A are it really mitigates the effects of maternal age, it increases implantation success, it reduces the risk of spontaneous abortion, it increases ongoing pregnancy rates, and it enables more efficient embryo transfers. Now, in the UK, we're governed by the HFEA, and over the last week or two, there's been some headlines around clinics offering false hope to patients and trading on hope. So Sally Cheshire, chairman of the HFEA, did an interview with the Telegraph where she was just questioning some of the decisions and how IVF may be being exploiting women's hopes. So I think it's very important that we do present the evidence to the patients. We work with you to ensure that you do understand the evidence around there for PGT-A, so we can justify this both to the patients and to the HFEA. So what can we do? And this is beyond just PGT-A, but basically we have to explain that there is no tool around that allows the perfect identification of the perfect embryo to result with a perfect baby. All we do is we have tools that can change the order in which the embryos are transferred to maximize the chance of a live birth for any transfer and lower the risk of spontaneous abortion. And we should only offer these technologies, whatever they are, and really here I'm talking about PGT-A, but it equally applies to other add-ons, that we offer proper counselling with the appropriate indication. And again, this is something where we as a company, CooperSurgical, are very keen to work with everyone. We have our own in-house genetic counsellors and embryologists and technology experts who can really help educate the clinics as to what to do. So we have improved a lot over the years, and basically we started off with morphology and looking more and more at embryo development, and then we started adding genetic testing. And genetic testing is one of the tools where we can use to rank embryos, and it's part of the incremental improvements. And we will continue as a company to look at how we can improve these incremental improvements to increase IVF success rates. And with that, I would just like to thank you all for your attention, and please contact us if you have any further questions with regarding this presentation or indeed any other CooperSurgical IVF and fertility products. So once again, I thank you very much for your time and look forward to hearing from you soon. Thank you.